Mitosis is the type of cell division in which a cell divides into two daughter cells, each with the same number of chromosomes as were present in parent cell. Mitosis occurs only in eukaryotic cells. In multicellular organisms, the somatic cells undergo mitosis. Prokaryotic cells undergo a process similar phases of mitosis. There are two major phases, i.e. the division of nucleus known as karyokinesis, and the division of cytoplasm known as cytokinesis, karyokinesis. The division of nucleus is further divided into four phases, i.e. prophase, metaphase, anaphase, and telophase. Prophase, normally, the genetic material in nucleus is in a loose thread-like form called chromatin. At the onset of prophase, chromatin condenses into highly ordered structures called chromosomes. Since the genetic material has already been duplicated earlier in S phase, each chromosome is made of two sister chromatids bound together at the same centromere. Each chromosome also has kinetochore at centromere. Kinetochore is a complex protein structure that is the point where spindle fibers attach. There are two centrioles close to nucleus, recall from chapter. Each centriole duplicates and thus two daughter centrosomes are formed. Both centrosomes migrate to the opposite poles of cell. Here, they give rise to microtubules by joining tubulin proteins present in cytoplasm. The microtubules thus formed are called spindle fibers. Complete set of spindle fibers is known as mitotic spindle. By this time, nucleolus and nuclear envelope have degraded and spindle fibers have invaded the central space. In highly vacuolated plant cells, nucleus has to migrate to the center of cell before prophase. The cells of plants lack centrioles, so spindle fibers are formed by the aggregation of tubulin protein. When spindle fibers have grown to sufficient length, some spindle fibers, known as kinetochore fibers, attach with the kinetochores of chromosomes. To kinetochore fibers from opposite poles attach with each chromosome. Chromosomes arrange themselves along the equator of cell, forming a metaphase plate. A number of other fibers from the opposite centrosomes attach with each other. Anaphase. When a kinetochore spindle fiber connects with the kinetochore of chromosome, it starts to pull toward the originating centrosomes. The pulling force divides the chromosome's sister chromatids, and they separate. These sister chromatids are now sister chromosomes, and they are pulled apart toward the respective centrosomes. The other spindle fibers also elongate. At the end of anaphase, cell has succeeded in separating identical copies of chromosomes into two groups at the opposite poles. Telophase. Telophase is a reversal of prophase. A new nuclear envelope forms around each set of separated chromosomes.